the Mendel, a very important part thing is the magic number 3:1. How did he arrive at 3:1? The reason why you have problem with the practical because you don't understand. I told you before. How did Mendel arrive at 3:1? What did he do? This is a magic number, right? This is called Mendelian. Mendelian inheritance. Am I right? Combined size also must not define this. Don't think not inside. When this Mendelian means?
Two heterozygous.
Heterozygous. I think a little bit more than that. It's about this one is referring the offspring. Told you before also is in the notes, right? Inheriting a pair of contrasting traits. Pair of contrasting traits basically you're talking about is the either black or brown eye color, for example tall or short pea plants, remember? So this is a top Mendelian inheritance. So this is what do you mean by contrasting traits? 3:1. How did you get 3:1? Come on. He did a few thousand experiments, right? More than 60,000. So he has a huge sample size. This is saying for the so many time already. So a huge sample size, correct? This is his actual, this is the theoretical ratio, right? We study Mendel's number because 3:1 is the magic number. So this is his theoretical ratio. Am I right? Correct or not? But how come if you do experiment, you'll never get 3:1? Seldom get 3:1 in experiments. First thing you must know, right? Because sample size too small. Sample size too small, right? Then this one will tell you that your what? Is not reliable. So in order to actually make up for the huge sample size, you really have to do a lot of times. So if not, it's not going to be reliable. This sense say a lot of times already, so this will be the last time I'll say it. Okay, the next one we want to look at is the what? The terms. Remember, there are a lot of terms that actually interact and they are similar to it one. Terms. The terms that we talk about first. What terms do we have? Come show me all the terms. Hello.
Genotype, phenotype, allele.
Okay, wait. Before allele got something. Gene, right? Allele.
Yeah.
Then genotype. So the terms are gene, allele, genotype, phenotype. Any more? Still have it. Add on, people.
Recessive, dominant.
Very good. Dominant, recessive. Who do we use this for? This one, right? Dominant, recessive, right? Okay, then some more. This one we got certain words can use one. Wrong. Only one can use. My bad. What words can I use for this?
Homozygous.
Very good. And heterozygous. Maybe this way, so you see the words that you are using, you should not crisscross and must be used in this way. Dominant, they ask you what is unknown by dominant allele. Guys, that the predictive don't know what answer. Sorry. What do you mean by dominant? Aiyo, already. Dominant is what?
Dominant, that means two.
It will express itself in
not sure.
what kind of genotype it will express itself? What kind of genotype?
Homozygous and heterozygous.
Yes, so when you are defining it, you need to write both. Don't stop here. Go on to say it's a genotype. Right? This Why we are doing this? Because they like to ask explain what is meant by. Remember? Nowadays you don't see define anymore, right? So okay, the next one. This one is what express itself in only. Am I right to say only? Correct, right? Homozygous genotype. The most horrifying thing about defining and putting the answers is that you reuse the words inside. Some of you will say because it's expressed itself in only homozygous genotype, and then you will start to put in the word recessive again. If you do that, right, then that defeats the purpose of trying to explain. You cannot use this word again, yeah? So should be like this, right? Homozygous genotype, then you know already. So this one can this one I'll use different color. This one can have what? Consists of only what? Only 2 dominant or 2 recessive alleles. Am I right? I know never asked before lah, but then you never know. You must understand it anyway. Hetero consists of, this one I use only because only this case only, right? Only 1 dominant and 1 what? Recessive allele. Food for thought, everyone, you must digest these 4 parts properly. There's no need to memorize it because you already know how it works, right? Just make sure you pair the correct word to the correct terms only. Any issue with this one? If you cannot do this properly, right, that means when you're asked to write answers whereby they don't allow genetic diagram for both the pure as well as the combined science, they can ask you to write in words. You cannot use the genetic diagram. A few of you asked me before whether can I draw a genetic diagram. Can, but if you use up your writing lines for genetic diagram, there's no marks for the genetic diagram at all if it's in the essay. So if it's just in words, you have to use these terms like how we are using it. Okay, so phenotype is easy. If phenotype is basically the what? Your turn. Hello people. Phenotype. Hey, I left the easy one for you. At the end. Hey grandma, 快点. Phenotype. Aiyo, I forget already. Hello.
The traits that's expressed.
Trait that is expressed, yes. If I make it more accurate, as a result of, you write yourself or later you watch the video. Later I put this on Flying Pigs as a result of what? You guess what would I write? Must think on your feet. I cannot help you all the time. Trait that is expressed as a result of the genotype right? Of the individual. Okay. They give you 1 mark, then you write like 1 mark Phenotype very difficult to be 2 marks There's nothing to write. This term must digest it properly. I know it may be a bit weird that the way I define it in the bank is a bit different, but there's another way of explaining This is another way of explaining it. Okay. Ash.
Can you also explain codominant?
Yeah, I'm coming to it. Hold on. Okay, 1 minute. Okay, next one. It's the important one, which is called codominance, right? The ones that we learned so far, there's only one example we discussed when you look at codominance. Co basically means what? You must first understand what it means for codominance first. So let's define the word codominance. Codominance means what? Two alleles in a genotype are, what's the word we use? Use codominant again cannot, because that is in the word. So they are what? Equally louder?
Fully expressed.
Yes, equally expressed. No, I don't like this one. Let me change a bit. Expressed in what? In its phenotype. Correct or not? Because phenotype can see, very obvious. Blood typing is the only one that we learned so far, right? The alleles are like that for blood typing. IA, IB, IO. You only have 3 alleles. These two are codominant. Okay, so they can go together and they express it as a phenotype, which is your AB blood type. Right? Okay? This is the only way you can get AB blood type. For IO, okay, IO basically this two is dominant over this one. Okay, so we look carefully now. So if it's A, B, and O blood type, what kind of alleles do we have here? A blood type. So when you write the big letter A, you're writing phenotype. If you accidentally go and do that for the allele, then it's wrong. The allele cannot be written as the big. So A, what kind of alleles do we have? IA. Sorry, wrongly. IA, right? Cannot. This is the genotype. This is the phenotype. Okay, so IA, any more or not? One more, right?
IAIO.
This one same thing. IB, IO. IO no O no choice. IO. Like this. IAB only have one. It must come from both together. So the genotype is IAIB. Can? Codominance, does it express 3:1? Does it express 3:1? Wait. Hey, hello. Does it express your Mendelian ratio 3:1? It need to think so long, man. Is it all the time you can express 3:1? When do you not see 3:1? Hello? When do you not see 3:1? We can get a normal, if there is a AB blood type involved, right? We will not be able to get this 3:1 ratio at all. You have done questions on this before, you know that you can't get Mendelian 3:1 if you have an AB blood type involved. Am I right? So you must bear in mind, unless it's complete Mendelian inheritance based on the front part we learn, right? Complete, okay, it means 3:1 will follow. This one cannot. Okay, codominance one, most of the time cannot follow 3:1. Do you know what I'm saying or not? Hello?
Yes.
Okay, so the writing part I leave to you. We go to the next one. The next one I always confuse one. This one I said last time already. I need to see again. I also don't know how. Come on. So please, variation. Take 3 still here or not? Run away. Did he drop out again? No, he's here. Okay, variation. Everybody must know this. Okay, variation. I told you there are 3, right? You must know. Number 1, that's the source of variation. Quick. Source of variation.
Genes.
What genes?
I don't know.
Which one? Next one is what? Crossing what?
Crossing genes.
Crossing over of the genes. Remember? Between who? Remember just now I say? Homologous chromosomes, remember? Correct or not? During which phase? Prophase 1, right? Prophase or meiosis 1. Remember this one, okay, please? So number 2, what's the other one? The source of variation. No, you have the independent first. The independent assortment. That means they line up independently of each other. Independent assortment of what? Homologous chromosomes. And metaphase 1. Correct or not? Third one, teacher must know very well. Teacher must know very well because you study this. Combined science with this one can use. So it's a random fusion of gametes. When? During fertilization. 没有啦 fusion. Fertilization, fusion. Hello. And there are two kinds of variation, right? You always confuse. So this is discontinuous.
Mr. Tan, the independent assortment is the pairing up of homologous.
After pairing, they line up at the equator. At the equator.
Then how they?
You remember just now that I got this? Just now I got this like that, right? Remember they were like that? Just now? Remember?
Yeah.
This is independent assortment. This one is the bivalent. Bivalent or the homologous chromosomes. Can or not? Combined science don't learn this, don't is okay. Who's still talking? Sorry, Danish. Hello. Danish talking, is it?
No.
Okay, can already, Okay, this one, continuous, discontinuous. Discontinuous basically is inherited, right? Right, you inherit this, and after that you inherit this, you realize that there are such traits have no intermediates. So when you plot it into a graph, you will only get like that. There's nothing here one. There's nothing here one. Nothing. You don't get anything here. There's no intermediate. So it's either black or brown eye color. For example, A, O, A, B, or B blood type also follow this. The continuous different, inherited. Alright, it's controlled by many genes also. Okay, and it's influenced by environment. So influenced by environment, so the graph you'll see like that, and they will give you a nice curve. So there are many intermediates in between. For example, if it's height, you will get 180 cm, you have 171, 162, 160, 141, 140, and then the list will go on. So you have a lot of intermediates. So these are the differences between these two you need to know. Are you clear or not? So make sure you know this one, and then the last one is the mutation, right? Mutation, okay. is this one, right? Mutation. Let's look at the mutation now. Mutation must be defined properly, and by now everybody should be able to. So mutations are what? Sudden.
Sudden, random, spontaneous,
Mm.
change in. I don't know what.
Wait, spontaneous. Change in either gene.
Change in structure of gene or chromosome number.
Correct. So you have to include two. Your definition must include two exactly the same one. Different. I mean, these two cases of mutation clearly. All right. And we will look at the example. Change in gene structure. Example that we learn is your sickle cell anemia. So sickle cell anemia, what is the cause of the disease? Change in gene structure means there is a change, or we say alteration, in the DNA nucleotides. No, wrong. In the sequence of nucleotides in DNA, eventually coding for what? Defective. Defective means not functional. Okay, not functional. What is that? Hemoglobin. Do you understand? It's a better way of writing it lah. Because the structure of the gene if it's a gene, the DNA is like that. A gene is here Okay, so if it's a gene structure change, it will be the sequence of nucleotide change. If the hemoglobin is not functional, this will lead to what? Decrease in oxygen carried by the red blood cell, right, to the rest of body cells. You should map it somewhere in your notes if it's not clear for you yet. So listen to the video again. So decrease in oxygen carried by the rest of the body cells, then what will happen? As such, you realize that your red blood cells, the RBC, will also have a sickle shape. And this sickle shape is no good. Why? They have a tendency to stick to RBC, have a tendency to stick together. When they stick together, they may block the lumen of arteries or veins. I know I developed a bit further for you now rather than the before, because if they ask you how do you discuss it, you need a number of points in order to get it across. So to phrase it properly, this is about the best way you can do it. Take some time and digest this part, okay? This is a little bit new for you, but generally the points are the same. Cannot. The cause is important. Chromosome number, the only one that we learn would be the Down syndrome. Right class? Down syndrome, easy. Cause 21st chromosome. 21st chromosome have an extra number or extra chromosome easy than that. So what will happen? The child effects child born with special features, or they may have a decrease in IQ or retardation possibly. This one not much to actually write about. This is just on the one of the two effects and usually the lifespan is also shorter. So if you map properly, the inheritance part you should be able to solve most of the questions other than the harder ones, but this one should be fine. Questions for this one, these are the main things involved in this lah. Natural selection you read on your own, okay? So combined science there is no natural selection involved, but mutations are all covered. Okay already? Everything okay? That's it, finish.